Welcome to today's webinar about circulating tumor cells analysis, a new workflow combining ScreenCell technology with Stilla Crystal Digital PCR. We are thrilled to have you join us for what promised to be a highly insightful session exploring a powerful new workflow at the intersection of cell isolation and high-resolution genetics. This cutting-edge approach leverages the strengths of both ScreenCell CTC enrichment technologies and Stilla next-generation digital PCR to advance the field of cancer research. This webinar will be presented by two outstanding experts who bring deep knowledge and hands-on experience in this rapidly evolving space. First, we have Lara Spitayou, a molecular biologist with a focus on drug and cosmetic bioactives. As field application scientist at Stilla Technologies, Lara specialized in digital PCR solution for high-resolution genetic analysis, helping researchers to achieve their goal with a simple workflow and high data quality. Joining her is Dr. Amira Bouzidi, a molecular biologist with extensive expertise in biomarker discovery, liquid biopsy, and molecular diagnostics. She holds a PhD in biotechnology and brings a strong background in cancer research. Currently, Amira leads R&D projects focused on molecular characterization of circulating tumor cells at ScreenCell, where she applies advanced tools such as digital PCR, NGS, and CRISPR. Together, Laras and Amira will guide us through this novel and simple workflow from efficient CTC isolation to in-depth molecular analysis, highlighting its potential impact on cancer for precision molecular testing. So let's dive, and please do not hesitate if you have any question during the session. Thank you very much and wish you a great webinar.
Hello, everyone. Welcome and thank you for being here today for this webinar. Where with Laras, we will present to you a new workflow combining ScreenCell technology with Stilla Crystal Digital PCR for circulating tumor cells analysis. Well, let's dive by presenting what's a liquid biopsy. So it's a minimally invasive assay performed on a sample of blood, urine, or other body fluid to uncover valuable information about the tumor from both primary and metastatic cancer sites. So a few examples of the liquid biopsy, it can be CTCs of circulating tumor cells as single CTCs or as clusters. It can also be circulating tumor DNA, microRNA, extracellular vesicle, etc. So these different components of liquid biopsy can provide important information about the tumor, about its heterogeneity, about the tumor subtypes, about copy number alteration, DNA mutation, etc. Initially, the standard for diagnosis and for clinical management of cancer patient is the tissue biopsy. However, we believe that liquid biopsy can complete this tissue biopsy because of its advantages. It's minimally invasive, it's highly sensitive, it takes short time. We can draw blood from patient at different time point of the disease, so we can monitor the disease and the response to treatment. And also it can reveal spatial and temporal tumor heterogeneity, which the tissue biopsy is quite limited for that. Our main interest at ScreenCells are circulating tumor cells. So what are those? They are cells detached from the tumors and diffuse into blood circulation. They can detach either from the primary tumor or from metastatic tumor, and they are also the only tumorigenic material remaining after tumor ablation. So after surgery or after treatment, the CTCs that can be still in the circulation are the only tumoral information left. So there are different interest and clinical application for these CTCs and for their characterization. First, they can help to detect early detection of relapse. There are studies showing if there are still CTCs in the circulation after surgery or after treatment, it can indicate a future relapse. It also allows real-time monitoring of therapeutic efficacy. So same principles, like there are still CTCs in the circulation, this might indicate a treatment resistance. Thanks also to this, the characterization of these CTCs can allow detection of therapeutic targets and resistance mechanism. A few words about our company, ScreenCell. We are a French biotech based in Paris. Our mission is to develop an easy-to-use, cost-effective CTC isolation technology for cancer monitoring. There are currently 70 scientific papers using our technology that are published, and we're involved in clinical studies on treatment efficacy, monitoring, detection of relapse, cancer aggressiveness, and all of this to have a clinical validation of our technology. So we have mainly two devices, the MB device for molecular biology and cell culture, and the Cyto device for cytology. It's a label-free, site-dependent isolation technology. So the blood mixed with the buffer will go through the device and will go through a porous membrane. On this porous membrane, you can see here on the right, the CTCs, which are way bigger than leukocytes, will be retained on the membrane, but also bigger leukocytes can also be retained, and all the rest will pass through the porous membrane. It's an easy-to-use, compact, single-use device. There's no need for an external instrumentation, and the whole isolation process takes less than 10 minutes. Also, our both devices are CE-IVD standards in Europe. Here are some performances of our devices. On the left, some internal data about the recovery rate when we spike cells. So if we spike 5 tumor cells from cell lines in blood, we will recover 91.2% when using CytoKit and 94% of them when using MBKit. And on the right, some scientific paper where they define a detection rate of CTCs using ScreenCell technology in different cancers. And you can see that from 85 to 95% of patients have CTCs detected on their blood. The CytoKit is useful for CTC enumeration and cytological characterization. So if you want to see the cells, their morphology and their cytological characteristics, like their markers, the markers they bear, this device is the one that you want to use. It's also compatible with immunofluorescence method, immunocytochemistry, FISH method, etc. With the MB kit, the cells are alive and unaltered after filtration. It's used for cell culture, so you can put this capsule here immediately in a well for cell culture, or you can also extract DNA and or RNA, and this DNA and RNA can be used for molecular characterization of CTCs and the detection of actionable and informative informations, which is the main goal of the workflow that we developed. So before detailing the workflow, let's define what actionable mutations are. So this is a representation of different alterations that are found across various cancers. Some of them are what we call actionable, meaning that they can guide treatment decisions. So some of them are responsible for sensitivity to treatment or the opposite, for resistance to treatment. So today it's a standard to test for these mutations, and the integration of genomic data, it improves prognostic and guide clinical decision making. So it is today important to look for the mutations in these genes for personalized therapy, and it also can be done on CTCs. Here are two examples of two genes that are targetable, that are actionable. So PIK3CA and KRAS bears mutations that can be targeted with the treatment. So PIK3CA hotspot mutation are activating mutation in breast, endometrial, cervical cancers. PIK3CA is a part of the mTOR pathway, and it's targeted by alpelisib. And here I give an example of a cell line that bears these mutations in PIK3CA, so it's NCF7. It's a breast cancer cell line, and this is the cell line that will be used in the workflow. So to mimic patient's blood, these cells will be spiked in the blood. On the other hand, we have KRAS gene with G12C mutation. So it's common in non-small cell lung cancer, colorectal, pancreatic cancer also, and it's targeted by sotorasib and adagrasib treatment. Oh, before explaining and diving into the workflow, we often receive the question, and also we can see that in the literature, what's the best liquid biopsy? Is it circulating tumor cells? Is it cell-free DNA? Is it something else? So there are some studies showing that there is a better detection and more specific detection of mutations in cell-free DNA. On the other hand, there are other publications showing the opposite, that there is a better detection in CTCs. So there is a discrepancy about that. You can also say that it's easier to analyze mutation in cell-free DNA because methods are more standardized, and the cell-free DNA do not need enrichment compared to CTCs. However, it's the CTCs that have a metastatic role, and also they are the only component that can track newly emerging resistance mutation under treatment. So the answer would be that these two are complementary. So it's important to test both of them to maximize our chances to find targetable mutation and to have a more holistic image of the tumor. Workflow that we developed with Stilla and combines ScreenCell technology and Stilla technology. We started by spiking cells of cancer cell lines, as I said before, to mimic patient's blood. We spiked H2030 cells or MCF7 cells, so we spiked different amount of cells in 3 ml of blood, and then the blood is mixed with a buffer, and blood plus buffer goes through the device, and then the cells are retained on the capsule. Here there is a stopping point, so you can store the capsules at -20, or you can carry on immediately with DNA extraction. For DNA extraction, we used QIAGEN microkit. The extracted DNA can also be stored at 4 degrees for a few weeks or longer at -20 degrees. And then we pursue with a whole genome amplification. So because after extraction, there is few amount of DNA obtained, which is normal because few cells are actually retained on the membrane. So a whole genome amplification is important to have more molecules to detect better later. The amplified DNA can also be stored at 4 degrees for a few weeks or -20 for longer. After that, we proceed with the Crystal Digital PCR. So we used both system, NICA and Nio system. We used Ruby chip and flex probes from Stilla. I'll hand it over to Laras, who will explain better the Crystal Digital PCR, the flex probes, and the results that we obtained.
Thank you, Amira, for your complete introduction. So yes, I will take over starting from this part. The digital PCR is a third generation of PCR-based DNA detection after what is already highly known classic PCR and quantitative PCR. So with the same elements of qPCR, including the fluorescent probes, the mix is first prepared in a tube before a run. So the first and the fundamental step in digital PCR is the partitioning of sample before amplification, which will allow individualization of the target and allowing after the fluorescence detection of the positive partition that contains the target after the PCR is finished. So how to move from positive partition quantification to the quantification of DNA is the central concept of digital PCR. It is from the principle that the term digital is used. So basically, we have a binary judgment between the number of positive droplets to the number of total droplets. With the help of Poisson statistic, the software will calculate the number of copy of our target of interest per microliter without the need of standard curve. So Stilla Technology is a French-born company that is specialized in digital PCR. We only do digital PCR as our core activity. We are the first ones to offer a 3-color digital PCR system on the market, continued with 6-color, and recently the first integrated 7-color digital PCR system on the market. We aim to be a complete solution provider in the digital PCR world. So with hundreds of happy clients all over the world. So today we offer you solutions that goes beyond instrumentation that I will explain in the next slide. The launch of our all-in-one Nio Digital PCR system. Last year we launched a probe technology, the Crystal Flix probe technology that will allow our user to enhance their flexibility in reaching high multiplexing capacity or the ability to detect several targets at the same time within the same sample. So just a quick introduction of the system used in the project with ScreenCell, the Nio Digital PCR system, which is an all-in-one dPCR system. There are 3 members of Nio family. The first is the beginner-friendly Nio Essential, and then the Nio and the Nio Plus that can cater higher throughput use with the possibility to do continuous loading. So according to your throughput need, you can run from 192 samples in the Nio Essentials up to 768 samples per day with the Nio Plus. So all 3 systems has 7 colors detection and all in one format with AI-powered droplet counts and also compatible to automated sample loading. So the Nio family offers a simple workflow that only needs one type of consumable, which is the Ruby chip. So following classic PCR sample preparation, the sample disposing to the Ruby chip takes around 5 minutes. User can put samples manually into the Ruby chip by monochannel pipetting or multi-channel pipetting. User can also use pipetting robot. These chips then will be loaded to the Nio, where partition and amplification takes place. The duration of this step will depend on the PCR program that is chosen by the user. The sample reading will take around 30 minutes, followed by the analysis. So the overall process from sample to result will require around 3 hours span. So with the ensemble of the ScreenCell technology and Stilla solution that we presented in the previous slides, we simulated the cancer mutation detection in blood sample. So now let's move forward to the experiment part. So as mentioned earlier by Amira, our gene of interest in the study is PIK3CA and KRAS. So PIK3CA is one of the most frequently mutated genes that represent predictive biomarkers for breast cancer. On the other hand, KRAS gene mutation is associated with poor survival of lung cancer. So to simulate the presence of circulating tumor cells in the blood, we spike 2 types of cancer cell line, as explained before, the MCF7 that harbors E545K mutation of PIK3CA gene, and then H2030, which harbors the KRAS G12C mutations in lung cancer. So for each category, we spike either 100, 50, 20, or 10 cells to 3 ml of blood coming from different healthy donors. So these are the bloods that are coming from non-cancer patient before proceeding to cell isolation with the ScreenCell MB device, DNA extraction, and also detection with the Stilla Digital PCR. So as mentioned before by Amira, an extra step that we did after DNA extraction is to do DNA enrichment with whole genome amplification kit. So with this step, we managed to increase the yield of DNA extraction up to 14-fold. As you can see here, without DNA enrichment, the mean of the obtained DNA samples is around 1.24 nanogram per microliter, while with whole genome amplification treatment, we obtain around 17.59 nanograms per microliter. So other than WGA, user can also enrich the extracted DNA with PCR-based allele-specific amplification that you can see here in this publication. You can see that the detection of PIK3CA E545K mutation will improve in the DNA enriched group compared to the non-enriched group. So in this study, we use Stilla, a readily available catalog assay, to be able to detect PIK3CA and KRAS mutation. As it is a simulated experiment with a known mutation, we use a simple format of this assay, which is a triplex assay that can individually detect either wild type E542K or E545K mutations, and also another assay from KRAS detection that can individually detect G12C. For your information, an extended version of the assays for each category of gene are also available to help you increase the detection capacity of PIK3CA and KRAS mutants. So all of the two assays that I showed you earlier is developed by Stilla Technology by using our latest probe technology, the Crystal Flex probe and Crystal Universal Reporter. So this technology represents our answer to the traditional Pacman probe that can be long to synthesize and expensive when it comes to high multiplex detection setting. So unlike the traditional probe that contained the target-specific sequence and the fluorescence reporter and the quencher, the Stilla probe technology consists of two different parts. The first one is the Crystal Flex probe that are non-fluorescent oligonucleotides that's specific to the target. They contain a non-target specific sequence that we call a tag oligo. The tag oligo can be released during PCR and then can trigger the fluorescence of their complementary Crystal Universal Reporter. So these are the steps that explain how the Crystal Flex Pro and the Crystal Universal Reporters work. So when the target of interest is present, the elongation of the oligos and also the 5-exonuclease activity of tag polymerase will help cleaving the tag oligo and release it to the buffer, to the environment. And then on the second step, this little tag oligo will activate the freely available universal reporter and give fluorescence. As the Stilla system offers 7-color detection, for each color of universal reporter, we have one specific tag oligo, so user can combine the personalized Crystal Flex probe part with the same pool of Crystal Universal Reporter. This, for example, different assays from Stilla that are compatible with the same set of Crystal Universal Reporter, as you can see here. The KRAS that has 3 colors can be used with the same pool of Universal Reporter as the PIK3CA that also has 3 colors for different application. So this can be used in a modular and flexible approach. For our DNA sample in this study, we use a non-diluted DNA extract as the VAF calculation is done as ratio of mutated copy to the sum of wild type and mutant copy. We use the maximum DNA volume, which is around 3.6 microliter in 5 microliter in a Crystal Flex probe settings. And then after preparation of the DNA and the buffer, we load it into the only consumable that we need, which is the Ruby chip. The sample loading in the Ruby chip only requires one piercing gesture without any sealing needed. It is already pre-filled with oil. In the video that you see here, this is how the partitioning is happening within one Ruby chamber. So there is no user interference to do the partitioning. So it's very easy and automated within the Ruby chamber. After the data is generated in the system, we analyze the data using the Nio Analyzer software. So each kit that I present to you earlier comes with the individual configuration for mutation detection that makes result analysis very easy and streamlined. You also have the possibility to visualize the fluorescence of individual detection, to have a control of the detection of a very rare target or a very low abundance target. So this is the result of the detection of the first group with MCF7 cell lines for PIK3CA E545K mutant detection. So as you can see, we can have a good detection of PIK3CA mutation with good repeatability throughout the replicates. For each group, we have around 6 replicates. You can see that CV value is quite low. So even though we only use 5 microliter of sample volume, there is no need of sample regrouping. The lowest number of spiked MCF detected is 20 cell in 3 ml of blood, and the mean of VAF percentage in this group is around 0.35%. Comparable sensitivity was also reached in the KRAS group. We still observe a nice repeatability within the individual replicates, and in this group we are able to detect as low as 10 cells in 3 ml of blood with the mean VAF value of 0.24%. You can also see that without the WGA treatment, we weren't able to detect any mutation at all. That underline the importance of WGA steps in this kind of analysis. So both results show how the combination of ScreenCell CTC isolation technology with their MB device and also Stilla Crystal Digital PCR is able to detect simulated CTC present in human blood samples, both for breast cancer and lung cancer cell line. We do observe a fluctuation in the value of VAF percentage that was observed. We see that there was no correlation between the number of spike cell and the percentage of VAF detected in this category. This can be due to several causes. We have a lot of leukocytes in human bloods, and we use blood samples from different donors, which has different age and gender, and humans can have around 4,500 to 11,000 leukocytes per microliter of blood, and there are small presence of leukocyte carryover as explained by Amira earlier in the isolated CTC. This can explain why we have this variation of the VAF observed within the different groups. But all this does not prevent us to have a good sensitivity of detection in our result. So with the ensemble of results from this proof of concept experiment, in this part, we just like to extend the perspective of the future use of this approach and present the interest of boosting further the multiplexing capacity. So as we know, cancer biomarkers are updating quite rapidly for different type of cancer. This is the example of the 2 cancers that are discussed within the studies. So each different biomarkers has their own importance and relevance according to the time of detection, whether it's going to be during the early prediction step, during follow-up of the treatment, or to verify the presence of resistance to therapy. So with our next generation of digital PCR, user can have the possibility to do these different settings. First, to do the routine testing with a midplex assay, for example, doing the experiment with different samples using the ESR1 18-plex assay, or do a throughout sample profiling with more than 50 targets assay by combining several different assays for one sample. So this can reach the level of detection that is comparable to NGS. So in order to achieve that kind of multiplexing according to the need of the user with different mutations, in Stilla we can push again the capacity of multiplexing by offering a color combination setting for your detection. In a traditional setting, one target can be detected with one color, but in color combination setting, we can tag each target with 2 colors that allows for 21 different color pairs in 7 colors. That can specifically detect 21 different targets. Of course, doing this with a classic fluorescence probe is costly. That's why using the Crystal Flex Probe and Universal Reporter can be a good solution for people who want to push their multiplexing capacity. So with Stilla, you can build your own pan-cancer panel according to your need using the previous method. We can give you assay-specific primers and assay-specific Crystal Flex Probe that can be combined with the Crystal Universal Reporter. According to your need, we can also give you a positive control for your experiment and even doing the LOB or limit of blank and limit of detection values for you to be able to use it readily. You can also choose from the readily available catalog your mutation of interest for a rapid detection of your application in different oncogenes. So you can visit our website to check the assay that are available for your needs. So with this part, I will hand the presentation back over to Amira to let her do the conclusion for the seminar.
Thank you, Lawrence. And so with this workflow, we showed that it is possible to detect mutations in CTCs using ScreenCell technology for isolation of CTCs and Stilla technology for crystal digital PCR when as low as 10 cells are present in 3 ml of blood. So a short wrap up about the workflow. We start with the screen cell filtration that takes less than 10 minutes, then DNA extraction, up to 1 hour, whole genome amplification that can take up to 4 hours, and then you finish with the crystal digital PCR, and in 2 hours you have the results. So the whole workflow can be done in 1 day experiment, depending also on how many samples you have. So it's less time, better sensitiveness than NGS if we compare to MGS, which is still the standard today. It's also cost effective compared to other manufacturers or compared to NGS. So this gain of time and of sensitiveness can improve clinical decision when you need to know if the patients have the mutation, the sensitivity to treatment or resistance to treatment. When you need this information, so this workflow can improve the clinical decision making for personalized therapy. And with that, we thank you for your attention. Thank you, Laras, for this joint work.
Thank you, Amira. We look forward to answer to your question. Thank you for your attention.
So, thanks a lot for this great presentation. I hope you all enjoyed the session, and we are open to answer all your questions and feedbacks. So we have a first question that I think I can answer to it. So nice talk, thanks. I have a question. Is it possible to do dPCR analysis from CTC in frozen sample? I have several frozen blood sample and would like to use them. So can we use the frozen blood sample from what I understood in the workflow for CTC extraction?
For now, our protocols are set up for fresh blood. Unfortunately, I'm not sure if the filtration will goes well when the blood is frozen. Yeah, we recommend fresh blood from EDTA tubes, or you can also use collection blood tubes with conservatives like Roche tubes, Transfix. This works well. We set it up. So these tubes can keep the blood longer in time, like till 48, 72 hours, and then you can do the filtration. But with frozen blood, unfortunately, I'm not sure that can work.
Next question, or message. So thanks for the talk. Will the presence of leukocytes in isolation skew the results and lead to missing very rare events?
Well, I would not say screw the results, but yeah, today there is no technology that can isolate exclusively only CTCs. You will always have a leukocyte contamination, but it can be an issue if you have very few CTCs, like we saw the results. If you have less than 10 CTCs in 3 ml, yeah, so the amount of the signal will be drowned in the leukocyte's DNA. But depending on the cancer stage, you can have nice detection even with the leukocyte contamination, I would say. Even it's not very contamination, it's always when you do an enrichment of CTC, you will always have some leukocytes left.
Okay, thanks. Next question, what is the sensitivity of CTC detection?
Yes, so when we do the filtration, we do quality control of our devices, and the sensitivity or the recovery rate is up to 90%. So when we spike 20 cells, we can recover up to 18 cells, and that's our criteria to validate our kits. What you mean by sensitivity, I guess the recovery rate, But about the sensitivity, what we show in our results, it's that we could detect for KRAS mutation, we could detect till 10 CTCs spiked, or for PIK3CA 20 CTCs spiked.
Okay, the next question is, what is the optimal blood volume? Will ongoing chemotherapy or targeted therapy impact the results?
The optimal blood you need to use is, as we did, is 3 ml, but you can go up to 6 ml of blood. However, if you filtrate 6 ml of blood, you have the risk to have more contamination with leukocytes, because there are way more leukocytes in 6 ml compared to 3 ml. So if you do in parallel, for example, cytofiltration, where you can determine the number of cells you have, let's say you found, I don't know, maybe 50 cells, 50 CTCs, then with 6 ml, you can do the filtration and detect the mutation in your CTCs. But the optimum, and to be safe, it would be 3 ml of blood filtration. Now, if the therapy or chemotherapy can affect the results, it depends on the efficiency of the treatment. It depends if the treatment gets rid of cancer cells. It's more of not a technical question, but more of a biological question. If there are some CTCs left after treatment, yeah, of course, we hope that the treatment will get rid of cancer cells and also of CTCs. But if with treatment you still have in the blood the CTCs, it can be a sign of resistance to treatment.
Okay, so the other question, so can you analyze RNA? I think it's an interesting question. Can you analyze RNA? The sensitivity may be much higher.
Completely, yes. With the advantage of the MB kit is that the cells are still alive, so it's good for RNA if they're still alive when you do the filtration. And yeah, you can do RNA extraction, absolutely. And yes, it can be better, I mean, depending on what your experiment for, but there are usually more mRNA molecules than DNA molecules, so maybe it will improve the sensitiveness. Yeah, completely.
And yeah, the same person asked also about the cost for a sensitive analysis of CTC, the price of the ScreenCell MB device, and also the price for the Nio device. I think, at least from our side, I would suggest that we get back to you directly with the information about your needs to really give you a specific answer toward your needs.
Yeah, I would just add, same, you can go on the website and ask for a quote, and also on our website, depending on what you want, what experiment you want to do and what is your project. We have two main devices, MB and Cyto, but there are different kits, like depending on the collection tube you use, depending if the cells are fixed, are they alive, you will get also advice to what kit to order.
For you, Amira, what is the pore size for your membrane? Is it something that we can share? Yeah.
No, it's fine to share. Yeah, the pores are 6.5 micrometers. Yeah.
Okay, and the second one still for you. I think the coming next question is for you. So, what is the purity of the recovered cells?
I'm not sure what you mean by the purity, but maybe the leukocyte contamination. Yeah, maybe. So yeah, there is always leukocytes retained on the filter, and as I said, there is no technology where you can have only CTCs and not leukocytes. But for the cytokit, for example, it does not affect the cytology method to see the CTCs, and in MB it can affect the sensitivity, but as we show in the results, you can detect till 10 cells depending on the mutation, 10 or 20 cells. So I'm sorry, I'm not sure what you mean by purity, but there is always a leukocyte contamination. Yeah.
Okay, great. The next one, do you capture any cells when healthy donor samples are used? If so, how many?
Yeah, so there is a limit of blank when you filter only healthy donor blood. So you can find some cells that have epithelial characteristics. Does it mean that it's tumoral cells? No, I mean, we cannot put a diagnosis on that. But if I remember correctly from a clinical essay we did, we put a limit of blank of I think 3 or 4 cells. I don't remember quite well, but it's in this area, 3 or 4 cells that are suspicious, we'd say, and going up from this, like starting from 4 cells, 5 cells, which are tumoral or have epithelial characteristic or have tumoral markers, we consider that it's positive for CTCs and start counting them. So yeah, there's always a blank, and in our clinical essays, we did filtration of a cohort of healthy donor, and we put this limitation of number of cells.
Great. The next question is, I think we can answer both for that question. What is the LOD of the assay for mutation detection? So I can answer for some part of this question, but I also leave to you. And the next part of the question, does WBC contamination in the isolated sample affect the WGA and further mutation detection? So I'll answer to the first part. So about the LOD. So the LOD will really depend on the sample matrix. So is it cell-free DNA? Is it CTC? Is it FFPE sample? So it will really depend. I think for this test, the LOD depends on the assay, 10 and 20 cells, if I'm not wrong, for what you were able to detect. For example, for PIK3CA.
Yes, it was. Let me check again.
Or maybe did you run an internal test to calculate this LOD?
Yeah, here we have for PIK3CA it's 0.3%, and for KRAS it was 0.2%. So actually, the experiment we did is it is to have a LOD limit of detection. So you go lower on the number of tumoral cells, and you see till when you can detect. Yeah. I don't know if that
And
answers.
the second part of the question, I don't have the answer. Does WBC contamination affect the WGA and further mutation detection?
WBC is white blood cells, I guess. It does not affect whole genome amplification. You will have in the filter, you will have white blood cells, leukocytes, and you will have your CTCs. Then you do the extraction, and in your extract, you will have DNA from CTCs and DNA from the leukocyte. Then you will do the whole genome amplification. The white blood cell will not affect. It's just the DNA of this leukocyte will be amplified too with the DNA of CTCs. And does it affect the detection or the sensitivity? I won't say it affects the sensitivity, but as you saw, we do not have control on what molecules of DNA, is it the CTCs molecules or the white blood cells molecules that are amplified and how much they are amplified. So that's one in the results. You can see that when we spiked 50 cells, you have a better detection than when we spiked 100 cells. It's also because of this whole genome amplification that can introduce not bias, but that can amplify Y type molecules. And yeah, I won't say it affects sensitivity. It does not affect whole genome amplification, but where it can play a role is if you have very few CTCs, these CTCs can be diluted. The DNA of these CTCs can be diluted in the Y type DNA, so the detection can be less effective. Yeah.
Okay, and the last question, is there a kit available for the isolation or detection of circulating cells in urine samples? Does those kits, for example, presented?
In our R&D department, we don't have a kit for the urine. Our kits are only for blood, but in the past, I know in our R&D department, we tried something on urine, but I'm not sure that it gives good results. Maybe contact us and we can see again what we did before, and we can talk about that. But yeah, no, for now, what we commercialize are devices for blood.
Great. Thanks for the remaining question. We will for sure give you an answer directly by email, so no worries. Thanks a lot for attending to this webinar, and we hope to see you in the next sessions. Thank you, Amira, for this great presentation. Thank you, Laras, also, and see you soon. Bye.
Thank you. Bye. Have a nice day. Bye.
Bye.